Neurodegenerative disorders. This is my summary of the Cognitive Neuroscience course. The diseases that I'll be covering include Alzheimer's disease and motor neuron disease. To begin with, in the case of Alzheimer's disease, the disease prevalence is only increasing since we have an aging population in not just Australia but in many countries as well. And the worldwide cost of Alzheimer's is believed to be estimated at $818 billion in 2015. 1.09% of the global GDP is affected by Alzheimer's. Early onset Alzheimer's disorder, or EOAD. This is when individuals develop Alzheimer's before the age of 65, and it may be sporadic, whereby there is no family history of this disorder, or it could be familial. Autosomal dominant. This is when a small minority have a mutation prone to Alzheimer's disorder, and it creates amyloid betas from PSEN1 to PSNE2 and APP, which is the amyloid precursor molecule or something along the lines of that. There are still lots of unexplained enzymes. Other than early onset Alzheimer's disorder, there is also late onset Alzheimer's disorder, or LOAD, which occurs in individuals who are greater than 65 years of age. Now, the affected brain regions of Alzheimer's includes damages in the cortex, whereby it impairs thinking, planning, and remembering. There is a shrinkage in the hippocampus, since it becomes harder to form new memories. As a result, the brain shrinks from Alzheimer's disorder, and this increases the size of the ventricles being the spaces between areas in the brain and where cerebral spinal fluid, CSF, flows between. Pathological Perspective. The amyloid hypothesis states that Alzheimer's derives from the growth of amyloid beta and tau proteins. Amyloid precursor protein, or APP, is a transmembrane protein which is in charge of cutting and making proteins in neurons. There is alpha secretase, or SAPP, whereby it is when APP uses this, as in the alpha secretase, to travel on non-amyloid pathways, and this is believed to be neuroprotective, whereby it stimulates neurogenesis, enhancing long-term potentiation, or LTP, and memory. Beta secretase, or SAPP beta, is when APP in disease goes through amyloidogenic pathways and through beta secretase. Eventually, in this pathway, there are waste products that build up, which are in the form of fragments, and these fragments cleave together to form amyloid betas. Amyloid betas are monomeric, which means that they exist as single molecules, and they are responsible for normal brain function in long-term potentiation and memory. But in disease, it becomes an oligomer, which is made up of two molecules, and this leads to toxicity in the cell and eventual memory loss. Fibril are when there are multiple molecules of amyloid beta, and this leads to development of plaque, whereby it is made of 42 amino acid amyloid betas. According to the IWG, two criteria in the diagnosing of Alzheimer's, Dubois in 2014 stated that Alzheimer's disordered individuals have specific clinical phenotypes, which is the early or significant episodic memory impairment that is tied with other cognitive behavioral changes, suggesting a mild cognitive impairment of dementia. There is a gradual progression whereby it occurs over the course of 6 months or more, and it produces impaired performance on episodic memory tests like acute recall, which shows the hippocampus is affected. In vivo evidence of Alzheimer's disorder pathology: a decrease in amyloid beta 1 through to 42 and increases in T as well as p-tau proteins in the cerebral spinal fluid indicates, or rather correlates with, Alzheimer's disorder. An increase in these tracer retention of amyloid in PET scans also can show the onset and prevalence of this disorder and Alzheimer's. And the Alzheimer's disorder autosomal dominant mutations are present, such as PSEN1, PSEN2, or APP. Amyloid PET scanning. This is used to detect amyloid betas prior to the onset of Alzheimer's disorder. It is controversial since some scientists believe that amyloid betas are not responsible and that they are not the main culprit of Alzheimer's disorder, since cognitively normal people have amyloid betas. And amyloid beta cascade testing found that it had 99.6% failure rate, since maybe the treatment was too late or the treatment isn't reaching the right brain regions, or maybe the disease cascade is completely false or wrong, as in our understanding may not be on par with what is actually happening. Neuroinflammatory cascade. This is when astrocytes and microglia become overactive, becoming very short or long, and injury may have started this disease cascade. This causes glial cells to become overactive and release pro-inflammatory cytokines, and thus they die en masse. J20 mouse model. The J20 mouse have inserted HAPP genes, which are 2 mutations, and they develop plaque buildup, and using a technique of SCC, it was found that the buildup didn't cause neurodegeneration, the buildup of HAPP gene that is, and that there was no neuronal loss other than initially in the CA1 region. There were increases in microglial in CA1 and CA3. Stereological cell counting. This is the SCC method that I should have mentioned before, and it is when neuroscientists count individual cells in the brain by placing them on a slide, and the cells are given traces. This is a very time-consuming task, and the count relies on random sampling, according to Wright et al. 2013. Radial arm maze. Amyloid beta mice are placed and tested via memory and learning tasks for food reward. Those with impairment, as in the mice that have impairment in information retainment after 24 weeks, are compared to controls, and you can see then that there are decreases in anxiety, but also, well, impairment, as mentioned before. There are decreases in anxiety in the elevated plus maze. Findings. So ultimately, amyloid betas are already present in the later stages of the disease. PET scanning isn't effective for pre-detection of Alzheimer's disorder. So what now? Well, other scientists have proposed that why don't we look into the blocking of AMPA receptors and how this could improve memory. AMPA receptor blocking helped mice to find reward, showing improved memory and learning. Given a reference memory test, mice were looked at. The short-term memory of mice were looked at over a period of time, and food was baited in each arm of the radial arm maze, and mice had to remember as well as to not repeat entering into each single arm. Alzheimer's disorder occurred before the plaque load, and treating amyloid betas is already too late. There is a need to treat neuroinflammation or neural cell loss to halt disease cascade progression, to halt the disease cascade. So yeah, that was Alzheimer's. Now I'll be talking about motor neuron disease. It manifests itself as Lou Gehrig disease or amyotrophic lateral sclerosis or ALS. It has a 3 to 5 year lifespan from symptom onset or rapid onset, and individuals retain memory and cognition but are unable to move, whereby they literally become trapped in their own body. You may remember the ALS challenge, the ice bucket challenge, which was used to help raise funds for this disorder, and famous people like Stephen Hawking have this, or rather had this disorder. Okay, there are 2 classifications for motor neuron disease. The disease could either affect upper motor neurons, which produce symptoms like increased tone, hyperreflexia, spastic gait, exaggerated jaw jerk, extensor plantar responses, and slowed movement. Or motor neuron disease could affect lower motor neurons, which brings about symptoms of muscle wasting, fasciculations, weakness, absent or reduce deep tendon reflexes, etc. The clinical diagnosis, as according to the revised Escorial criteria, states that evidence of lower motor neuron degeneration by clinical electrophysiologic or neuropathologic examination, whereby the upper motor degeneration by clinical examination have progressive spread of symptoms within or to other regions. The study of motor neuron disease requires a multifaceted approach, whereby psychologists, or in this case neuropsychologists, look at familial history, neuroimaging, physical examination, electrodiagnostic studies, and lab testing. Pathological perspective. The spinal cord has white matter atrophy in motor neuron diseased individuals, whereby there's a loss of motor neurons in the anterior horn of the spinal cord and motor cortex, and these have neuroinflammation. Some scientists have found that there is ubiquitin positive inclusions, which is similar to amyloid beta plaques, which are observed in neurons of the frontal temporal cortex, hippocampus, and striatum. It was found that the gene or protein TDP43 in 2006 was involved in ubiquitized inclusions in the ALS. Motor neuron disease is mostly sporadic, occurring without a familial history of it in 90% of cases, and that being the 10% of cases being familial, as in genetics spread from one generation to the next, and it involves mutations like the SOD1, which affects 20%, mutations like the TDP, which affects 3% of all cases, and mutations like the FUS affecting 5%, and C9orf72 affecting 38%. Riluzole or Rilutec is a drug that increases the lifespan of individuals with motor neuron disease, but it doesn't reduce symptoms of it, and all it does is act on sodium channels. TDP43 research by Wright et al. Relied on mice who were fed with the protein docs, and this carried the mutation of HTDP43 delta NLS, which mimicked motor neuron disease. By 10 weeks, the mice died due to motor neuron degeneration, and we could already measure the waning or the loss of grip strength by 3 weeks. That is 3 weeks after being injected with the protein/mutation. Neuroinflammation occurs in these mice by 2 weeks, and in 4 weeks you can see microglial and expansion of microglial populations, since there is neuroinflammation occurring. Quantitative qPCR. This takes RNA out of the motor cortex and exponentially doubles it. After 2 weeks, scientists can see 16 genes that become upregulated before neuron decreases, as in before it dies. 4 weeks afterwards, 14 genes are upregulated, and then 1 gene is downregulated, and then it produces neuron loss. You can see how genes peak and start to decline in the inflammation process, and genes could be either pro or anti-inflammation, as in they could either increase the amount of inflammation happening or try and reduce it. CXCL10 is a gene that stands out, and the protein ELISA test is used to measure levels of CXCL10. It was found that CXCL10 increases 11-fold. CXCL10's role in motor neuron disease. It is measured through immunofluorescent staining and tagging of primary as well as secondary antibodies with fluorescent tag. TDP34 mice had similar inflammatory markers in humans. There needs to be more research into whether this protein helps or hinders neuroinflammation and can either reduce the amount as well as see the effect of this protein on behavior and in our mice model. Similarities between Alzheimer's and motor neuron disorder include neurodegeneration and neuroinflammation, though they don't happen in the same areas, they don't affect the same areas, and other similarities include that they are affected by proteins like amyloid betas, but these proteins are not the same thing. So Alzheimer's is affected by amyloid betas and MND is affected by TDP. Another similarity is that protein drives the disease. So yeah, thank you for watching. We looked at Alzheimer's disorder and motor neuron disease, recent research into this field. Thanks. Bye bye.